nervous system. The nerves are going to give you the peripheral nervous system. The brain and spinal cord, that's going to be the central nervous system. If we scroll down, you have this division in boxes: central nervous system, brain and spinal cord, peripheral nervous system, nerves. We have two types of nerves: the ones that are going to make you feel, right? The ones that are going to tell you that you were bitten by a mosquito, and the one that is going to tell you that you need to move to scratch it. Nerves that are going to make you feel, they're going to be sensory division. And all those nerves that are going to make you move something, they're going to be called the motor division. The nerves that help you move the muscles that are attached to your skeleton, that's going to be called somatic motor. The ones that make you move your heart, visceral motor, because they're inside of you. Sensory, they're going to make you feel. So if you got bitten by a mosquito, then you know that's in your skin. But do you feel pain in your intestines when you have diarrhea, for example? Of course. So then how do you know that you have pain? Because you have receptors for pain in your intestines, and that's why you know that you have diarrhea. So how many sensory you have? You have sensory somatic and sensory visceral. How many motor you have? Motor visceral and motor somatic. Motor visceral has two additional divisions, which are going to be the sympathetic and the parasympathetic. Okay, let's keep going. The neuron. The neuron has the body, all this part right here, and it has extensions. These extensions that are smaller, dendrites. This long extension is going to be called axon. This axon is covered by this balloon type of structure, kind of like blue right here. These are going to be called myelin sheath, as you can see here. The spaces in between these areas is going to be called the node of Ranvier. Classification of neurons: if it has multiple extensions, as you can see here, multiple extensions, it's called multipolar. Bipolar, how many extensions? Two. One right here, another one right here. Which one is this one? If it has only one extension, unipolar. Okay, the next topic is supportive cells. We said that the cells that you find in your brain are going to be the neurons. Well, in addition to those cells, there are other cells that the main job that they have is just to support the job of the neuron. For example, if there is an infection in your brain, the neuron doesn't fight the infection. Other cells will do that. So these other cells that you find in your brain are going to be called the neuroglia. As you can see there, you have the neurons and how these other cells are around these neurons in order to give them support. That's the neuroglia. Oligodendrocytes form myelin sheath. We have ependymal cells. They produce the cerebrospinal fluid. Microglia, they fight infections, for example. Astrocytes, they are part of the blood-brain barrier. So we said some of the functions of these cells is to produce myelin. Myelin is very important for the speed at which the information is going to be transferred from your body into your brain and from your brain into your body. So the myelin wraps around the axon, as you can see here. This provides you myelinated and unmyelinated neurons. So this one is myelinated, where you see the Schwann cell, how it wraps around the nerve. That's myelinated. This one right here is considered unmyelinated. The main difference, one of them is faster than the other when it transfers the information. Which one is faster? The myelinated. Which one is slower? Unmyelinated fibers. The nerve fibers, they regenerate by just a tiny, tiny bit, which is not enough in order to repair or regain the main functionality on some areas where the movement of the muscles has been lost. Electrophysiology, resting membrane potential. In the case of the neuron, is -70. The main ions involved on this are sodium and potassium. Most important ion outside is going to be sodium. Most important inside is going to be potassium. Once you receive the stimuli, sodium channels open. The sodium goes from the outside, extracellular, inside to the intracellular area. And because of that, the polarity is going to change, right? It's going to change from -70 to +30. Once +30, potassium will come out, and that will return the cell back again to the resting potential. Now we have local potentials. So local potentials, they start, but they don't reach the trigger zone. Therefore, those are weak stimulus. They can be excitatory or inhibitory,
but they do not trigger an action potential.
And right here you have a table where you can compare action potential and local potential. The characteristics for local potential, they are decremental, right? They start, but then they don't reach the trigger zone. They are graded. They are reversible. They can be excitatory or inhibitory. Here are characteristics of the action potential. This is all or none law, meaning once it starts, it starts. You cannot stop it. Okay, non-decremental, The stimulus doesn't decrease in reversal. The action potential will continue until it reaches the end of the neuron. Refractory period. Right here you have the action potential again, depolarization, repolarization. Absolute refractory period. It's called absolute refractory period because you have one stimulus right here and another stimulus cannot happen at the same time. There's something else called relative refractory period. Okay, relative refractory period refers to this part. You see the yellow column right here? Okay, this is negative 70. This is the resting potential. However, as you can see here, when the potassium starts coming out, so you can reach the negative 70, a lot of times the channels keep open even though you already reached negative 70, so the potassium going out overshoots the negative 70 and goes even beyond that. So let's say this point is negative 80, more or less at this level. The theory says that if you have a stimulus that is very strong, you can start an action potential right here. You will start it in this yellow area. Therefore, that's why it's called relative refractory period, because you can initiate it even though it's not negative 70. If we scroll down, the next topic that we have is signal conduction in nerve fibers. We said we'll have two main types of fibers. We are going to have the myelinated and unmyelinated fibers. This unmyelinated, you see there's no myelin sheath covering the axon. Inside is negative, it needs to become positive, right? Sodium goes inside, so everything goes from negative to positive. In order to do that, you need to open channels for sodium, and that allows sodium to go inside. So let's say that from here to here, there are 100 channels for sodium, and you need, let's say, 1 second to open each channel. So then how many seconds it will take you to take the information from here to here? Since you have 100 channels, then it will take you 100 seconds, because you need to open every single channel from this point until the end. So then, how long does it take you then to go from this point to this point right here? It's going to take you 100 seconds. Let's take a look now at the myelinated fibers. You have right here the axon. The neuron is negative. Now channel for sodium will open and sodium will go inside. However, but most of the channels are going to be covered with the myelin sheath, which is this. So the only channels that are going to be open are only these channels that you find in the nodes of Ranvier, which are the spaces between each set of myelin sheath. So let's say that we have 10 channels. So then, in this case of the myelinated fibers, we need to open 10 channels only. Since we said that we need to use 1 second to open each channel, it's going to take you only 10 seconds. Why? Because you need to open only 10 channels. And 10 seconds is faster than 100 seconds. In conclusion, the myelinated fiber transfers the impulse way faster than the unmyelinated fiber. Since in this unmyelinated, you need to open every single channel, then this is called continuous transmission. In the case of the myelinated fiber, since you need to open only the channels that you have in the nodes of Ranvier, so this one is called saltatory conduction. Okay, the next topic is synapses. Similar to what the neuromuscular junction is, in the case of a synapse, is a communication between a nerve and another nerve. Cessation of signal. So how do you stop these signals from continuing transmitting? You're going to take back the neurotransmitter into the neuron. That's a reuptake. In some cases, the neurotransmitters get released, but they reach areas where there are no receptors, so they just get reabsorbed and destroyed. And the other one is degradation at the level of the synaptic cleft. Like in the case of the acetylcholine, there's an enzyme in there called acetylcholinesterase that is going to destroy that particular neurotransmitter. Structure of neurotransmitters. When we talk about the neuromuscular junction, we said that the nerve is going to release acetylcholine. We can consider that the neurotransmitter acetylcholine is excitatory because it's going to make you move your hand. But you have to understand that it cannot be that the same acetylcholine is going to make you stop your hand moving. So that means that there are different kinds of neurotransmitters. Some of them are going to be excitatory, some of them are going to be inhibitory, the same way the gas makes your car move and brake fluid makes your car stop. Neurotransmitters, there are different kinds, and they fall under the category of acetylcholine, amino acids, monoamines, or neuropeptides. GABA, for example, is inhibitory, so that's going to make you stop. And we said acetylcholine is excitatory. Neural integration means that you have a lot of neurons and they work together, but everything is coordinated. If you have 80 neurons that are going to be positive, one of them is going to give you one stimulus, the second one, another stimulus, the third one, another stimulus. Little by little, you start adding up whether that's going to be excitatory or inhibitory. You just continue stimulating the summation. 1, 2, 3, 4, 5. Eventually, you're going to get enough in order to get an action potential. The next topic is neuronal pools. It's a pool because the neurons, they work in groups. So the first one is divergence. So for example, you have light, and with light, you will be able to see with this set of neurons. You're going to be able to maintain equilibrium with this set of neurons, and this set of neurons are going to tell you that you are awake. Converging, for example, this is the nerves that are going to help you move your diaphragm. When you inhale, these nerves are going to help you inhale and you move your diaphragm. But in order to exhale, you use these other nerves, and that's going to help you move your diaphragm. Reverberating, using the same example of inhaling and exhaling. This one is inhale, exhale, inhale, exhale. You need to go back in order to trigger this process again. So that's what is called reverberating. Parallel after this charge, you step on a piece of glass, you say ouch with this one, and you move your feet away from the piece of glass. These two things happen at the same time.